Okay, so enzymes are biological catalysts. Okay, now what that means is that an enzyme's job is to get a reaction started. Okay, the enzyme's job again is to start the reaction, inject some energy into the reaction, and it does this, as you're going to see, by lowering the activation energy. It's like having a good partner. An enzyme is like having a good partner. A good partner is supposed to decrease the amount of work that you as an individual does, not make you work harder. Okay, if you're working harder and you have a partner, then that's not a very good partner. Okay, so enzymes make the reaction go faster and occur at a faster rate. Otherwise, the reaction would be very slow. And so they're very important in living things. Again, enzymes are known as biological catalysts. Let's look and see what it does here. Okay, so we've already analyzed this graph before. We see the amount of energy needed. That is called the activation energy. It wasn't described in the last lecture, but it's described here. It's the activation energy. Very important to know that the solid line is the normal reaction that will occur without an enzyme, and we have the dotted line to show us what will occur with the enzyme being present. And what do we see? We see that with the enzyme present, what happens? It decreases. The amount of work needed for the reaction to occur is less. Okay? So the reaction rate goes faster because there's not as much work to be done, and in the end result, the reaction is catalyzed appropriately. Okay? Without presence of an enzyme, then this reaction would be slower. Now, who knows what that reaction would be in the body or in the bodies of a living thing or organism. Okay, that sentence, obviously, enzymes allow chemical reactions to occur under tightly controlled conditions, should be in the left-hand column. And what this means is that it is very specific. Okay, one type of enzyme is only going to work on one type of reaction. They're not going to work on different reactions. They're very tightly controlled, very specific to whatever reaction that needs to be done, but they can be reused, okay, as long as the conditions are right. So that means, what does that mean? Right pH, the right temperature, the right amount, okay? All of that stuff is very specific, and that's what it means when it says tightly controlled conditions. Has to be optimal, okay? They don't like variation. Enzymes don't like variation, and they're not very adaptable to change. Okay, so the conditions have to be right in order for an enzyme to work. This is very important. We learned this yesterday with our chart. Enzymes fall into the protein category. Okay, so that was one of the examples that was in the chart that we used yesterday that you're going to have to remember for quiz and test coming up. Most reactions, if not all reactions that occur in our bodies, are dependent on enzymes. Even the ones we don't think about or can control, like our heartbeat, our blood flow, our temperature. All those things are things I are not able to control on our own, but the enzymes catalyze those reactions. So again, mentioned some of this on the previous slide. A break in homeostasis. Remember, homeostasis is balance. Optimal conditions. Our bodies work very well with even keel, even balance. Temperature too high, temperature too low, not good. Okay? Human beings generally, our internal operations work best when it's balanced. Even nature, when it's balanced outside, when you have enough predators, enough prey, and a variety of different organisms in the community, the natural community, homeostasis exists, and that's good. Okay? So, right pH. Okay? Enzymes work very well. If you change the pH, you change the temperature, you're going to break the hydrogen bonds that attach these things, and also the enzyme's function. Size, shape determines function. Structure determines function in the case of an enzyme. If it cannot fit nicely into the substrate, which you're going to learn, which is part of how it works, then the enzyme will not work. It's like having a key that fits that lock right there, or a glove that only fits a certain size hand. If it does not, and when you put the glove on, if it doesn't fit properly, it's going to be uncomfortable. If you put the wrong key into that door, you're not getting in. Okay? Wrong enzyme is not going to work with the wrong substrate. Okay? They are very specific. Again, continuing what I said here, we have our enzyme working right here. We have our substrate. If these two things don't fit properly, no reaction is going to occur. If something happens, a chemical, a drug, anything that changes this shape, then the substrate and the enzyme will not be able to connect, and then the reaction will not occur. Okay? So anything that changes it. A lot of our reactions and things that happen in our bodies are a result of enzymes and substrates working together to produce a result. So a lot of the drugs and medicines that we take help control, stop, start a reaction in our bodies. Okay? And so sometimes if we take a drug, a recreational drug, it gets in the way of a natural reaction and takes the place of that natural reaction, and then what's supposed to happen doesn't happen a lot of times. Okay, so again, enzymes, substrates, and active site. The active site is the place where the enzyme and the substrate do their work. Mentioned this before. It's like a lock or a key or a hand to a glove. Substrate, enzyme work together. The bonds are weakened. The enzyme release and the reaction is carried out. Now, guess what? As long as the conditions are optimal or correct and right, that enzyme and substrate can be used again. Okay, it can be reused and reused and reused. The product is produced. The reaction in the enzyme, the substrate can work together again.